Okay, thank you guys for being here tonight. I'll go ahead and get the evening started. First and foremost, my name is Brett and I'll be your host and moderator for this evening, so thank you all again for joining us tonight. A quick format about what will happen tonight is that, that's my radio. Quick format is that I'll do this quick introduction. Thank you, Amber. Caitlin and Adam will come up and they'll speak for roughly about 45-50 minutes, and then we'll do a quick little intermission if you guys need to get more refreshments or need to use the facilities, and then we're going to come back and do an extensive question and answer and discussion session. So I want to just say, to remind you guys that tonight our event is part of the Global Health series that is funded by the Bill and Melinda Gates Foundation, as well as our contributing partners, the Washington Global Health. And so the series is part of my Science in the City program, and these events highlight the amazing individuals and organizations in our region who are working on innovative, ambitious, and scalable solutions to address health problems that have a major impact in developing countries. So again, thank you for that. Also a reminder that if you are a member with us, these events are free. Luckily, with the sponsorship through the Bill and Melinda Gates Foundation, these public ones for Global Health are free already. But if you're a member with us, all Science in the City events are free. So something to look into. If you do need to leave at any time, restrooms, other facilities, we do have a concession stand open outside if you need something to eat. Do that through the left-hand doors here on my right, so left for you. They're camouflaged, wood doors, head outside. So down the hallway outside, restrooms are going to be downstairs, concessions are just going to be there. When you re-enter, please do so through the doors that you guys came in through originally. So our next Global Health and Development Series lecture, so it's monthly, so the next one is on September 11th at 7 p.m. and it will feature the I left them in here. So our next one is actually going to be with Emilio Imani from the Bill and Melinda Gates Foundation. He's director of HIV and AIDS over there, and he's going to be talking about a kind of a personal reflection on the AIDS epidemic and where we are heading in the future. So I am looking very forward to this one. In addition, we have Intellectual Ventures sponsored a series with us. It's called Innovation for Good. And that one is a 3-part series that started in July. And so we're going to have another one here at the end of August. And again, this series explores impact invention. And so each lecture will feature a local innovator who is developing practical technological solutions that address real-life issues in collaboration with those who need it most. So our next lecture for that is on August 28th at 7 p.m. and it will highlight researchers who work on biomolecule detection for use in conservation and health realms. So it is my pleasure tonight to introduce our two speakers, Caitlin Beaver and Adam Okulian, to talk about Portrait of an Epidemic, Mathematical Modeling in Modern Day Epidemiology. So Adam is a postdoctoral fellow at the Gates Funded Institute for Disease Modeling, an affiliate assistant professor in the Department of Global Health at the University of Washington in Seattle. Adam has a PhD in epidemiology from the University of Washington and a SCB in environmental science from Brown University. As a member of IDM's research team, Adam focuses on spatial, mathematical, and epidemiological modeling of HIV in sub-Saharan Africa, with the goal of understanding transmission dynamics in high-burden settings. And so Caitlin is a senior research scientist at IDM, specializing in malaria and sleeping sickness in sub-Saharan Africa. Much of her work is devoted to figuring out the right recipe of tools, such as drugs, vaccines, and bed nets, necessary to eliminate these diseases in different countries. Caitlin has a PhD in biological engineering from MIT and a Bachelor's of Science in Physics and Astronomy from the University of British Columbia. So join me in introducing or bringing both Adam and Caitlin up to the stage tonight.
Okay, so just a few technical notes before we get started. I'm going to pre-apologize if I goof up using the slide laser device, whatever this thing is called. And also, we are going to be asking you guys some questions. And if you have any burning questions while we're talking, you're also free to ask us. Okay, so. Any questions yet? All right, so first question, show of hands, who here has heard of the Ebola virus? Right, everybody's heard of Ebola. So if you've been paying attention to the news, there's actually 2 new outbreaks in the past few months in the Democratic Republic of Congo. But probably most of you became acutely aware of Ebola during the panic around the 2014 outbreak that started in Western Africa. So what I'm going to show you now is an animation of the data that was collected during that outbreak. So you're going to see cases show up as colored pixels on this map. And we're also going to keep count of the number of people who have died as a function of time. So you can see the outbreak started in Guinea and spread quite quickly to neighboring countries, Sierra Leone and Liberia. And then eventually you have a few cases popping up in the US, in Europe, as well as some other countries in Africa. So I think the simulation actually doesn't go quite to the very end of the epidemic because there were over 11,000 people who died in this epidemic. So that's very bad. But there are certain properties of Ebola that prevented the situation from being much worse. So one thing to keep in mind is that Ebola virus is actually not all that transmissible. You actually have to be in pretty close contact to somebody to get Ebola from them. And the other thing that's really important to know is that by the time you're contagious with Ebola virus, your symptoms are really, really bad. So you're not likely to be running around in the public spaces, right? I mean, it happens, but not very often. So what we started thinking about was, what if the Ebola virus had different properties? What if, for example, it had similar properties to the Spanish flu, which caused a global pandemic in 1918? So the Spanish flu is much more transmissible because you can catch it through airborne particles. So the next video I'm going to show is a simulation of what would happen if the outbreak had started in Western Africa, as it did with Ebola, but now it has the properties of the Spanish flu. You ready for this? Okay, so we went from a situation that was very bad to a situation that was much, much worse. So what do we do to help prevent this kind of catastrophe? I'm asking you questions now. Anybody, what could we do to help the situation from getting this bad?
Vaccinate.
Right, so we could work on a vaccine. So you have to have some idea of what kind of vaccine it is that you have to develop before this happens, because it's hard to develop them quite quickly, but that's an excellent idea. What else?
Isolation and quarantine?
Yes, isolation and quarantine. Excellent.
Universal precautions.
What do you mean by that?
In the case of flu, maybe you wear a mask if you're in a public place.
Perfect.
Shut down international travel.
Yes, that's right. Prevent people from traveling. So this got as bad as it did because people are going all over the place. Right. But so how do we know which of those approaches is going to be the most effective? Should we just, run an experiment? Should we drop a virus in the middle of Seattle and see what happens? No. Yes. You came to the right talk. So there are lots of epidemiological questions like this that we can't test in the real world because it might be very expensive to do that, or it might be really dangerous to do it, or it might be just downright unethical. And that's exactly the type of question that we answer at the Institute for Disease Modeling.
So welcome to our world of disease modeling, where we are behind the safety of a computer screen as opposed to being in the field. My name is Adam Akulian. I'm a postdoctoral fellow at the Institute for Disease Modeling. We were started 10 years ago to work on malaria eradication, and so We're funded by Bill Gates, the Global Good Fund, to specifically develop mathematical models that can help to understand what sort of interventions would be most effective to eradicate infectious diseases. Our modeling started with malaria and expanded to many other disease areas, and we have grown to about 80 individuals, part software engineer and part researcher from many different types of backgrounds. So what is disease modeling? Well, we are not modelers in the sense that you might think of, runway modelers. We are a group of epidemiologists, mathematicians, computer scientists, software engineers, but we're also not working on the front lines of epidemics like you might have seen in the movie Contagion. We do things more like sit around and wonder what's driving epidemics on a whiteboard. So this is Caitlin and I yesterday pretending like we don't know what's going on, which actually happens a lot. But we do have a lot of fun, and this is a photograph taken 2 or 3 years ago. I had an opportunity to visit Western Kenya, where the HIV epidemic is very severe. Got to meet some really fantastic young boarding school individuals, these boys here, high school age young men who were involved in HIV prevention. And so we do get to have real experiences with people who are directly affected by the epidemics in which we are working, and then we get to come home and get behind our computers and think about developing models that will help eradicate diseases.
All right, so what is it that we do when we go to sit down at our computers to do this computer modeling of disease? So first step for us is to think about all it is that we know about the disease that we're interested in modeling. So this could be anything from understanding the biology behind the disease. It might involve understanding the environment in which the disease takes place, and also understanding the populations that are affected. So once we feel like we have a pretty good understanding of all the things, all the elements that play a role, we have to translate that knowledge into math. And then we have another step which involves taking that math and translating that into language that the computer understands. But once we've done that, we have this computer model of the disease. Essentially what we've done is we've taught the computer how to think about the disease for us. Because oftentimes there's so much going on in an epidemic that you can't keep all that information in your head. So we need a computer to answer these questions for us, such as questions related to learning from epidemics that happened in the past. So if we look at, say, measles epidemics that have already happened, maybe we can learn new properties of the disease. We can forecast the future of an epidemic, say that's already happening. So, while the Ebola outbreak is happening, we can make a prediction about how many cases we're going to see at its worst. And then we also do a lot of answering questions at the level of evaluating hypothetical scenarios. So again, this is very much related to what we just discussed with Ebola, which is, for a particular disease, what is the best approach for us to take in order to solve the problem? So Adam and I are going to give a couple examples of the work that we've done at IDM in the HIV and malaria spaces around evaluating such hypothetical scenarios.
So my work revolves around understanding the epidemiology of the HIV, the global HIV epidemic. So some of these slides might be familiar to some of you. I'm hoping that some will be familiar and some will be new. But I wanted to give you a picture first to start off with of the current, or this is the 2015 depiction of HIV prevalence by country in the world. So what jumps out at you about the global HIV epidemic from this map? Africa. Correct. So anything else?
South.
Right, so Southern Hemisphere, is the epidemic evenly distributed? No. So that's one of the first things you'll notice in a lot of infectious disease epidemiology modeling is we actually kind of take advantage of the heterogeneous distribution of diseases to understand, well, why is it so bad here in Southern Africa whereas there's not really an epidemic in other places. And actually Russia is having its own epidemic right now, and that some of these countries actually are not filled in because of lack of data. But that question has been posed over and over again. Why does Southern Africa have such a terrible epidemic? And I think we'll hopefully answer some of those questions now, and if those come up later, you can ask them. But the take home is that Southern Africa experiences a disproportionate burden of HIV in terms of its population size. This map is taking that other map and then distorting it according to the population that is infected with HIV and redrawing the map's lines to depict that. So you can see that Southern Africa is really experiencing a disproportionate share of the epidemic, and it's really bad. In parts of South Africa, upwards of 30 to 40% of the adult population is infected with HIV currently. So this is a current snapshot of, well, this is from 2013, but this is pretty similar to what it is today in terms of the prevalence of HIV and AIDS in the general population of adults. And so you can see over time that in South Africa, the number of cases of HIV, the number of current cases, the number of cases of HIV has been growing. It's starting to level off now that we have treatment, but it is continuing to grow over time. And so I wanted to give a little bit of epidemiology 101 for the people who haven't experienced or haven't been exposed to some of the terms that I'm going to be using. But the difference between prevalence and incidence. Oftentimes, if you read in the New York Times, they oftentimes get these two measures of disease wrong. And so if there's one thing that you learn from this, if you can learn the difference between prevalence and incidence from this talk, I'll be very, very happy, because then you can go home and you can correct your mom or your grandfather or your child or whoever, your dog who's ignorant about epidemiology. So I'm hoping that this slide depicts sort of the relationship between the two. So prevalence is the current snapshot. So if I were to take a picture right now of the HIV epidemic, how many people are currently infected right now? So it's prevalent infections. It's people who, so that's going to include people who have been infected for a really long time, and then people who just got infected yesterday. That's prevalence. Incidence is, so prevalence measures burden. When some people say the burden of disease, they're referring to the prevalence of disease, and usually we measure it as normalized to the population, so it's a percent. Incidence, on the other hand, is a measure of risk. So incidence is the rate of new infections. So when you hear incidence, you should be thinking about new infections. So what is the incidence of HIV today? Well, in certain countries in Africa, it's about 1 per 100 people are infected newly every year. 1 per 100 people per population. And so you can see the relationship between prevalence and incidence. So when you have new infections, it's like water flowing into a bathtub. The faucet, when you turn on the faucet, the more you turn it on, you're upping the incidence and you're increasing the flow of new infections into the pool. Prevalence is the pool. And so there are different things that can change how prevalence increases and decreases. So what is a way to increase prevalence? So if you increase incidence, if you turn the water up, the pressure of the water up, the water will rise. What about draining the bathtub? So if you increase the death rate, what is that going to do to prevalence? It's actually going to drive it down. So this seems counterintuitive, but in the HIV epidemic, we have incredible treatment that has prolonged the lives of people living with HIV, that the prevalence is actually not declining. And that's actually a good thing, because we're keeping people alive. And so if the prevalence is dropping because people are dying, that is not a good public health measure. That is throwing the baby out with the bathwater, no pun intended. But there's another way to decrease prevalence. Cure people. Excellent. So if we had a cure for HIV, which we don't currently, then we could cure people with HIV and take them out of that pool. Okay, prevalence and incidence. So now when you look at this slide and you see, oh my, the prevalence of HIV is continuing to grow up, or oh my, it's not changing, you can actually be rest assured that part of the reason that this is not declining is because we're keeping people alive with HIV for a long time. So now to some HIV biology, just some simple HIV biology, because another term I'm going to be talking about is viral load. So viral load refers to the amount of virus that is in a host body. So when you're initially infected, you can see that there's time is on the x-axis, and then we have viral load, which is measured as number of virons or copies of virus per milliliter of blood. So initially, when you're first infected with HIV, just like when you're first infected with any virus, there's a spike of virus in your blood. Your body is not able to detect the virus, it's replicating rapidly, and you have this spike of virus in your blood. At some point, your body actually responds to the HIV virus. It develops antibodies to the HIV virus, and those antibodies are actually what determines a positive test. So you're actually tested. A lot of HIV tests you for the presence of those antibodies to the virus. And so once you start developing those antibodies, the virus starts to crash because your body is doing a really good job of fighting the virus. But at some point, the virus is tricking your body, it's mutating, and it's able to escape your immune system, that eventually the virus will come back to a point at which it will cause AIDS and lead to the crashing of the immune system and finally death. But with the advent of antiretroviral therapy, and I'll refer to antiretroviral therapy as ART from now on, countless lives have been saved, because what this essentially does is it prevents the virus from replicating. It is able to insert itself into the replication process of the virus, into the HIV is a retrovirus, so it actually uses your DNA to replicate itself. It's able to halt that process, and so you can see, here's a depiction of an individual at 3 years, they got tested, they got put on treatment, and then their virus essentially went to 0. Well, it doesn't actually go to 0, it goes to an undetectable level, but it's always there. And because of that, individuals can live now a healthy lifespan if they are put on ART. And this is from the San Francisco Chronicle from the 90s, AIDS deaths cut in half, and it was no longer on the top 10 list in the US, burden of disease. And so this has been a miraculous discovery, the discovery of HIV treatment. But the second discovery that no one really thought of at the time is that, and this was the breakthrough of the year in 2011, is that if you treat people, you can actually prevent infection. And that wasn't actually thought of. It was thought of more as, let's help the people who are already infected, let's help them live healthy lives. But they thought, well, if you're driving the virus down, then maybe we could actually prevent further transmission of infections. And so that's why this was considered the finding of the year. It was written up in the New England Journal of Medicine. They found that individuals who were put on ART when they were having unprotected sex with their partners who were negative, very few of those partners acquired HIV. And so how does this work? This treatment as prevention, it's called. Normally, if you have an infected gentleman on the left, that person's virus is replicating inside their blood, and if they're having unprotected sex, they can transmit, I refrain from using another graphic, but this is not actually how HIV is transmitted, this is just a cartoon, just FYI. But they can transmit HIV easily because they have enough virus in the blood. If you're sick with the flu and you cough in your friend's face, that you're exposing your friend to virus. But the way we have thought about preventing someone from getting HIV in the past is by protecting the negative partner. So we don't have a vaccine right now. We're hopefully testing a vaccine right now, but there are pills. There's PrEP right now, so pre-exposure prophylaxis. It's a pill you can take every day to prevent HIV. And then there's also things that have been around for a long time, like condoms. So traditionally we think about prevention like preventing the person who is negative from getting it, but there's also the breakthrough of the year, where we can give the HIV positive person a treatment that lowers the copies of virus in their blood to such an undetectable level that it prevents transmission. And it's really a profound concept. And it's something that has sparked the interest of a lot of HIV mathematical modelers and epidemiologists. So now it's no longer about, it's not only just prolonging life in those who with HIV, but it's also about preventing transmission. And this concept has been tested. So this was a study done recently of 1,000 mixed status couples. So there's this concept of serodiscordant couples. So couples where one individual is positive and one individual is negative. When you put that positive person on ART, you can see in this study that there was 58,000, this is a study, so they documented this, 58,000 sex acts without a condom. So this is where the negative partner was potentially exposed to HIV, but there were 0 transmissions of HIV when the partner was on ART. So this was a breakthrough, and soon after this study came out, these slew of studies, the global HIV community adopted the idea of treat all. So the idea is if you can test and treat a substantial enough portion of the population who has HIV, then you could end the epidemic. So the question that was posed to modelers, so people who wanted to test this out before actually rolling out the intervention, because it was going to be a while until this could actually be could be seen. They wanted to know, answer this question: could we end the epidemic by treating all, by rolling out this concept of treat all? And this is one of the earliest studies. This is the mathematical modeling study from 2009, published in The Lancet. They simulated 3 scenarios. They simulated a scenario. I'm going to take this out. Yes. Oh wow. Stand up. Okay. So they simulated 3 scenarios. One in which there was no intervention, so they just simulated it, and as Caitlin said, you can do that with modeling because you're not dealing with real people, you're dealing with ones and zeros in computer binary. So they simulated the epidemic under no intervention. What would happen? This is what would happen to the epidemic if we didn't do anything. And then they simulated with certain thresholds. So we're going to treat people whose immune system have gotten to a certain point, and then we're going to treat them. That's the blue line. And then they said, okay, what would happen if we rolled out universal voluntary HIV testing and immediate ART? Because before, you couldn't get treatment until you were really sick. But now they were saying, well, let's treat people before they get sick so we can prevent infections. And you can see the epidemic crashes. It goes to essentially 0. And so this paper was published in 2009, and it got everyone really excited about the idea of test and treat. And it's gotten into popular media. This was published in 2017, so a glimpse of hope for beating the epidemic. And in this article, they postulated, based on some shaky evidence, but they postulated that if you could test and then treat 73% of people who are HIV positive, that is the tipping point. So it's like, who wrote The Tipping Point? That was a popular book. But you could reach this point at which you've essentially not vaccinated, but you've treated enough people that the epidemic will burn out. And so this was the beginning of the end of AIDS, and there was a huge push to get everyone tested and treated, especially in places like sub-Saharan Africa, where the HIV epidemic is the worst. And so you can see here's Barack Obama getting an HIV test back in 2011, and so this idea was being promoted and tested. And so they actually wanted to actually test this in the field. So they knew that it could work theoretically in a controlled experimental population, but could this work when rolled out in the field? And so they did this massive study where they tested and treated one set of villages and didn't do anything, or did standard of care in the other villages, and they wanted to see whether the incidence went down in the treatment communities relative to the control communities. And they actually found that incidence did not change. Incidence was not different between the two. It blew everyone. It was very disappointing, and they basically showed that this wasn't working. And there's a number of reasons why it didn't work, and I'm going to get into some of the reasons. It didn't work in this setting in Southern Africa because people weren't going on treatment. They were testing a lot of people, but they weren't linking them to treatment effectively. And so the intervention theoretically can work, but when put into practice, there's a lot of messiness when you actually try to implement something. And so, as mathematical modelers, part of our job is to also understand, well, what is the best way to implement a certain intervention that we know works? Because we know test and treat is good, and it should be implemented, and it should work. And so one of the studies that I'm working on right now is to understand the effect of test and treat in the country of Swaziland, the Kingdom of Swaziland, which is a tiny landlocked country in southern Africa. It has one of the worst epidemics in the world. These are the 4 provinces of Swaziland, and you can see the HIV prevalence is almost 30% among the adult population in these places. But Swaziland is also doing an incredible job of test and treat. So if you look at this map right here, it shows the percent of HIV positive individuals who are on treatment. Is this the prevalence or incidence? Good, it's the prevalence of treatment. So prevalence can be anything, but it's the prevalence of people who are currently on treatment. They took a snapshot and they said, what is the proportion of people who are currently on treatment? So what is the prevalence always has a numerator and a denominator. What's the denominator? Who's in the population here? Yeah, HIV positive adults, good. So this is, very good. We passed Epidemiology 101 with flying colors. That's the denominator, so it's people who are HIV infected. Among them, what proportion of them are on treatment? And you can see that there's upwards of 73%. So it's not 100%, but it's 73% of people with HIV are on treatment and theoretically virally suppressed. So they are not able to transmit HIV. And according to that New York Times article, that was the tipping point. Remember that, the 73%. So Swaziland should be on their way to ending the epidemic. And so we wanted to test that in our model. And a little bit of background on the HIV epidemic in Swaziland. And this slide is actually reflective of the epidemic in a lot of Southern African countries and Eastern African countries. The age distribution of HIV. What do you notice about, there's 2 things you should notice. What do you notice about the pattern of HIV prevalence? So remember, this is prevalence. This is people who are currently infected. Okay, more females. This is true. The prevalence is higher in women than it is in men. And certain age groups, do you see this? So 20 to 24-year-old age group, it's like 5 times as high in women as it is in men. And I'll get back to why that is. But what else do you see? Older. So the older you get, so let's talk about the age. Why is it that there's more older people infected? So yeah.
Just because you've been around longer, you have more chance to get infected.
Very good, excellent. It's prevalent infections, and so we're sort of building up a pool. But then why does it drop? People are dying, right? So as you get older, people are differentially dying faster with HIV than they are without, and so the prevalence actually goes down in the older groups. Excellent. And now the gender discrepancy, because this is actually seen everywhere in sub-Saharan Africa. Women have higher incidence, they also have higher prevalence. They also have lower mortality. They also have lower mortality. So that would also, what does lower mortality do to prevalence? Increases it, right? So prevalence is not all bad, but it's not a good thing because it's the burden of disease. Okay, so remember this picture because it's going to play into the next slide. This is the data that I'm using in my model. So this is a chart of the scale up of ART over time, so treatment over time. We're looking at HIV positive individuals here. We're looking at men here. This is just an example. But these are real data. So from 2005 until 2016, ART has been scaling up over time. What's something you notice here? Yes?
The older you are, the more likely you are to be being treated.
Why is that?
Because you live to be old.
Great. So there's a selection bias here. So the longer you live with HIV, the more opportunities you're going to have had to be on treatment. And if you didn't get on treatment, what happens? You probably have died. And so you see this, and this is kind of true across the board. Younger people are less likely to be on treatment. So you can see that back here. So what does that mean for the epidemic? That's a really tricky question, but what is the effect of having more older people on treatment than younger people?
Well, it's being spread when they're younger, isn't it?
Very good. Okay, we're starting to do research and understand, well, who's spreading HIV, and if you remember from the slide from the beginning, how viral load changes over time, this has part of the answer to the question of what is the effect of not treating younger people or not having the same level of treatment in younger people. Well, it turns out that a lot of transmissions, a disproportionate number of transmissions per unit time occur early. Why is that? You can see the virus peaks very early on, and this is an unfortunate aspect of HIV and a lot of viruses. This is why they're able to persist for so long, is because by the time they're found, let's say a year later, someone gets tested, put on treatment, a lot of the transmissions have already happened, and it's a sad fact. And so the only way to overcome this is to try to get people on treatment as soon as possible, but it means getting people on treatment very soon. And this is a question we don't totally know, and this is actually something we're modeling, is how soon do you have to get people on treatment to end the epidemic? So this is basically a translation of that last slide into the number of infections. And so I just went over this, but over time, theoretically, there's more infections that can occur early on. And if you treat a person at time t, that's not supposed to say secondary cases, that's supposed to say time. At time t, at a certain time, if you treated them there, well, all the red time has been, all those infections have already occurred. So there's no way of preventing those infections. Okay, so I have just a couple more slides and I'm going to turn it over to Caitlin. But one of the things we can do with modeling is to do things that you could never do experimentally in the field. And one of those things is to say, well, what would happen if we denied treatment to people? Or what would happen if we scaled up ART in a certain way? So the way I modeled this is to try to reflect the actual scale of ART over time in Swaziland, this incredible scale up of treatment, and then maintain that level as one scenario. And then the other scenario is to just look at what would happen if we didn't treat anyone. And so the results are fairly self-explanatory and expected. The orange line, this is time on the x-axis, and then we have prevalence on the y-axis, and incidence on the y-axis in the second chart. But if we just look at the chart on the left, you can see what happens to prevalence if no one's treated. And then you can see what happens to prevalence if ART is scaled up to what it's at right now, which is about 80 to 90%. So you can see that prevalence, it doesn't reach as high of a level as if we didn't treat anyone, but it maintains itself. So that's that pattern that we saw where prevalence is not declining is actually expected because we're keeping people on treatment. Incidence, on the other hand, is really what we're talking about, which is new infections. We want to prevent new infections, and you can see that there's that crash in new infections over time, and that has already happened. Actually, from 2000 to 2015, we've seen this dramatic decline in incidence in Swaziland, and that's actually true. The data confirm this, that the HIV incidence in Swaziland has been cut in half over the last 10 years. And we can theoretically show what would have happened if they didn't do the scale up that they did. With that orange line, that's the incidence, the number of new infections, the rate of new infections, had they not treated people. But did they end the epidemic? Could they end the epidemic? And I simulated this epidemic out to 2050. And so you can see that while the incidence does go down, it doesn't go down to the levels that would be expected. So the incidence is still 0.5 per 100 person years. That's how we measure incidence, person years. So it doesn't go down to that 0 level that we would want. So the last slide, overcoming challenges to universal test and treat. When you implement something that works perfectly in the lab, you don't get the results that you might expect because societies are messy and there's still a lot of stigma out there. People are not getting treated as quickly as possible. There's biological reasons for why test and treat is not as effective as originally thought. There's also the age heterogeneity that our fellow colleague over on the left, or on your right, alluded to. So what we're really worried about now is the people who are not on treatment. And so one of the biggest goals of the current epidemiology and epidemiologic modeling, and then also interventions, is how do we get those people who are not on treatment, and what is their contribution to the epidemic. Next is getting people on treatment early enough so that you can prevent those infections. And then setting new treatment targets. So maybe we should update our treatment targets, not only 73% overall, but what about 73% of the young people? What would happen if we did that? And that's actually a modeling exercise that I'm doing right now, is asking the question, well, what would happen if we got those young people up to where the older people are? And what would be the cost of that? And what would be the effect of it? And then there's always prevention. So test and treat is an amazing invention and concept, and should be continued, but there's also developing new technologies and ways to prevent people from getting HIV in the first place by protecting the negative individual. And with that, I'm going to turn it over to Caitlin, who's going to be talking about a different disease.
Okay, I'm really not sure if I can handle two things in my hands, but I'm gonna try. So, now we're switching gears a little bit to talk about malaria. So, there are a few important ways in which malaria is different from HIV. First of all, malaria is not caused by a virus, it's caused by a parasite. And that parasite is transmitted to humans by... Yes, mosquitoes. This one is almost too beautiful, but I'm not really a fan. So people who carry malaria infections, they have parasites in their blood. A mosquito will come along and bite them, and as it's taking up this person's blood, it's also taking in the parasites. The parasites go through a maturation process within the mosquito's body, and when the mosquito goes to bite somebody else, they inject them with parasites, and they get malaria. Okay, so I was kind of interested to know if people in the audience, they leave the lecture and they go home like, I'd like to learn more about malaria, and they go to Google and they search for malaria, what are they going to find? So, there's this oh gosh, see, I knew this was going to happen. Okay, now the joke's been given away, but you have this this very handy little summary box that provides some details about malaria. But what's something that you read there that's a little bit strange? Very rare, yes. So it is true that malaria is very rare in the United States. However, that is not really true globally. I feel like this is a little bit misleading. And actually, they've had this up here on Google for several years, and I did write them a very diplomatically worded email explaining that I thought this was a little bit misleading, but they did not answer me. And it's still there. Okay, so malaria is not rare if we have a global perspective. In 2016, there were more than 200 million cases of malaria. I mean, that's a really big number. So a lot of the cases are in sub-Saharan Africa, most people know that, but you also have a lot of malaria in South and Central America, in Southeast Asia, and also in India. And we have made a lot of progress in the malaria space over the last 10 years, but over the last couple of years, this number hasn't actually changed a lot. And something that's especially concerning is that we still have a lot of annual deaths from malaria. In 2016, almost 500,000 people died from malaria, and the majority of those are children under the age of 5. So malaria is biologically quite interesting because it's something that you can acquire immunity to over time. So if you're somebody who lives in Burkina Faso, as an adult, you're very likely to be carrying parasites, but not to exhibit symptoms. because you've been exposed to parasites your whole life, and your body learns how not to get symptoms to that. But children don't have that advantage. They haven't had a lot of exposure to the parasites, so they don't have this immunity. And also, children just don't have the same kind of immune system. Their constitution is not as strong as adults. So when they get infected with parasites, they're much more likely to get symptoms and to get even a severe form of the disease. Okay, so it seems like we've got a really big problem on our hands. But there are a lot of people who are very optimistic that we may even be able to eradicate malaria completely. So the WHO, the World Health Organization, for example, has a goal to reduce malaria burden worldwide by 90% by 2030. That's only 12 years away. That's pretty impressive. And Bill Gates has stated that he wants to see the eradication of malaria within his lifetime. So, I think these are pretty smart people. Are they crazy? No. So, there are some good reasons that they're optimistic. And that's because for malaria, we actually have a lot of very helpful tools to help us combat the disease. So, when we think about eliminating malaria, there are a few different classes of approaches. So, one thing we do is a set of approaches called vector control. So vectors are the mosquitoes. So we do a bunch of things that are either going to kill the mosquitoes or prevent the mosquitoes from acquiring parasites. So there are probably some of these approaches that you have become familiar with if you read the news. So bed nets, right? Hands up. Who's heard of bed nets as an approach to, right? Okay. Everybody knows you can use bed nets to help solve the malaria problem. If you want to impress your friends, you can use the technical term long-lasting insecticide-treated nets. So these work in two ways. So obviously, if you're sleeping under a net, the idea is that you don't get bitten by mosquitoes. So that's handy because not only do you not get malaria, but you don't get bitten by mosquitoes, which are really annoying. And then they're also treated with insecticide. So a mosquito lands on the net, bad things happen to the mosquito. So similarly, we use indoor residual spraying. So this is kind of similar. You take similar insecticides and you spray them on the insides of homes, in schools or other buildings. Mosquitoes like to hang out on walls, so they come in your house, and they sit there, and if your wall is treated with insecticide, very bad things happen to mosquitoes. So this is kind of an aside, but actually some of the insecticides paralyze the mosquitoes, and their legs get all stiff, and their legs fall off, and then they can't really do mosquito things if they have no legs. And I read this paper, which was kind of disturbing, but also super interesting, where they deliberately took legs off mosquitoes and saw whether they could do mosquito things. And they did have some superhero mosquitoes who could mate with one leg. I don't really know, but anyway, that's just interesting. So the other class of things that we do to eliminate malaria relate to humans. So my colleague the other day reminded me that there's a very popular joke at malaria conferences. I don't mean we should get rid of the humans. That would work, but that's not really what we're going for. No, we want to get rid of the parasites in humans. So, for malaria, unlike HIV, we actually have drugs that can cure you. It's amazing. You take the drugs, they get rid of the parasites. So somebody had this great idea that we could do mass drug administration, or MDA, which is you go door to door in a region and you give everybody drugs so they get treated at the same time. So everybody clears their parasites all at once. But there's another approach related to the human side of things, which is not quite as exotic, which is this idea of just making sure that people have access to healthcare. This is very akin to what Adam presented to us about HIV. So when you are symptomatic with malaria, like when you have a fever, that is when you are at your most infectious. So if you can encourage people to go get treatment as soon as they feel sick, you can do a whole lot to reduce malaria transmission. So I've highlighted here this Munyumbwe Health Center, because this is a place that we know pretty well at IDM. So Munyumbwe is in Zambia. Here it is in Africa. Here's a closer look at Zambia. One of the reasons that we're interested in Zambia is because the Zambian government has a fantastic program to try to get rid of malaria. So they have very ambitious goals to eliminate malaria from their whole country by 2021. So that may or may not happen, but they're really, really trying very hard. And they're partnering with a lot of international organizations to figure out the best way of doing that. And the Gates Foundation is one of those organizations, PATH, which is also sort of in this neighborhood, and at IDM, we're also working on this. One thing that is especially interesting here is the story of Lake Kariba. Anybody heard of Lake Kariba before? So Lake Kariba is actually a man-made lake. It was created when they dammed up the Zambezi River to create power in roughly 1960, I think. Okay, so that's great, create power. But what do you think happened when you flooded a big area in Africa with water? Mosquitoes. They're like, we love this, thank you. So people living around Lake Kariba got lots of malaria. But this also makes it a pretty interesting area to study, because it means that the area around Lake Kariba actually doesn't have a lot. And so it's a great place to test how you would get rid of malaria without worrying too much about importation from the surrounding area. Okay, so here is a snapshot of the lakeside. So Lake Kariba is there on the right, and you can see we go up into the hills. Each of these circles represents a household, so where people are living in the area. The color of the circle is representative of the prevalence of malaria. Anyone want to take a guess what prevalence of malaria means? I'm sure you answered it correctly. Yes, it is the fraction of people in that household that are infected with malaria. So you can see clearly down by the lakeside, things are looking pretty rough. They sort of get a little bit better as we go into the hills. So this is where the Munyumbwe Rural Health Center is. So it's in a place where malaria is not too bad, but this is the catchment area. So when we talk about catchment areas, that's the area that defines basically the border within which everyone is supposed to go there for their health care. So I know there's no scale on here, it's sort of a weird angle, but this catchment area is sort of, I don't know, 20, 25 kilometers across. And if you squint, you can also see this is a pretty rough geography. So here's a couple of pictures. This is what it looks like. It's quite beautiful, but it's very sandy. There are almost no roads. Those hills are actually some pretty serious hills again. So it's really hard to get around. So what that means is that some people living in Munyumbwe are going to have a really hard time getting to the health facility if they get sick, right? Especially if you're sick. If you feel terrible, are you going to walk 3 hours to get some drugs for malaria? Well, you might if you're worried about dying. If your child gets sick, yes, you will probably make the effort to take them there. But if you're an adult and you just feel a little bit bad, you might not. So that's an issue. So people are not going to be accessing healthcare if all they have is that health facility that's really far away. So in Zambia and a number of other countries, they come up with this idea of community health workers. So these are essentially volunteers who get a little bit of training from the Ministry of Health to be able to test and treat their neighbors, essentially, for malaria. So most of them are volunteers. They may get a small stipend, and it's incredible. I mean, we're going to see with some simulations what the impact can be. Okay? So I'm going to show you a simulation of the Manyumwe Health Facility catchment area. So each of these dots here is representative of the prevalence, yes, of the people living under that dot. So I'm going to start the simulation assuming that people can only get healthcare at the facility, which is somewhere around here. About halfway through the simulation, I'm going to introduce a dozen community health workers. So with the facility in place, some people are going to have to travel really far to get care. With the community health workers in place, almost everybody is going to have a point of care nearby. Okay, so let's start the simulation. Now you see flickering colors. That's telling you that the prevalence is changing over time. And there's a lot that's going on here that you can't see. So you may notice these like major changing trends in colors. That's because in the model, we have seasonality. So we have rainfall and temperature that's changing over the course of the year, and that is driving the amount of habitat that mosquitoes have to lay eggs. And when mosquitoes emerge, they have the chance to bite people. Those people may or may not be infected already. If the mosquitoes do acquire parasites, they can go on to bite somebody else. And we model the demographics of those people that they might bite. So they could be adults, they could be children, and those people are going to have different levels of immunity to malaria as well. Okay, so we're near the end of the simulation. Did anyone see when I added the community health workers? Right, no, probably not. You have to have really keen eyes to see when the community health workers were added, because community health workers don't solve the problem all at once, right? So community health workers solve things incrementally. You're not clearing the parasites from everyone all at the same time. You're encouraging people to seek care, and that gradually over time reduces the risk of every individual in the area. Okay, so if we add up the numbers, actually, we have the risk of all the people just by adding the community health workers. This is pretty darn amazing, right? Remember, these community health workers are almost free. So just by helping people have access to get care, you go a really long way to solving the problem. But we haven't totally solved the problem. Remember, Bill Gates, he wants to get rid of malaria completely. So this is not going to fix everything. Anybody want to guess why this doesn't completely solve the problem? At the back, yes.
The lake is still there?
Well, yes. So that's a fair point. So you still have mosquitoes here. So if you have anybody who's infected, it's possible that they can create onward transmission. But there are also some reasons around the fact that, okay, imagine that you're sick. Do you go to the doctor? No. Right. A lot of people are like, oh, I'll power through it, it's fine. Even if you lived right next to your doctor's office, you might still say, no, it's all right. So that's an issue here. You have people who get sick, and even though you've made it easy to access care, they're not going to do it for whatever reason. And
Are there any non-human reservoirs? Would it just move into some other animal?
So that's a very good question. It depends on where you are. So malaria is not generally thought of as a zoonotic disease. But there are diseases where that matters a lot. Other vector-borne diseases, like sleeping sickness, for example. So there's another really important reason why this doesn't completely solve your problem. And that's really, I saw a hand, go for it.
People get reinfected, or when they're adults, they're infected asymptomatically, so they're not going to do that.
That is exactly right. So remember, adults have this immunity, so they're getting parasites, and they don't feel all that bad. So you have all these people walking around carrying parasites, and they don't even know it. But they can still transmit the parasites to other people. So what should we do instead? Well, Zambia said, let's take all the tools that we currently have in our anti-malaria toolbox. Remember, we talked about nets, and we talked about spraying and mass drug administration campaigns. They said, we are going to throw the malaria kitchen sink at this problem and see what happens. So this next simulation is a simulation where we include all of those different interventions. Anyone want to guess what happens? So I'm going to run this much faster this time, so you can. Oh. You know what? I turned this upside down. Okay, let's try this again. Okay, so we see the seasonal changes of malaria. It's going up and down with the rains. Eventually, they start adding in these interventions. So they add the community health workers, and they add the nets, and they add the spraying, and they do mass drug administration campaigns. And after a few years, you almost completely get rid of malaria. Well, I mean, this is a simulation, but actually they did this, and this is more or less what happened. They haven't quite gotten to zero. You'll see there are a few pockets of places where there's still malaria, but they've gotten very close. So this is very, very exciting, because I have not talked about any of the magic bullet approaches to malaria that you might have read about in the news, like genetically modified mosquitoes. This is, purely the unsexy approaches that we already have and know work. And so essentially, though it may be very difficult for us to completely eradicate malaria with the existing tools, we should be able to get very close to elimination in a lot of places with what we've got. And that is definitely, I think, reason for hope. So, I turned upside down again. I did warn you. It's symmetrical. That's a very important. All right. So that actually concludes the research portion of our talk. But we are going to discuss a little bit about IDM, and of course, I guess we're going to do an intermission. We can talk a little bit about IDM. You can ask questions while we're talking about it. Yeah.
So there's many ways that you all can get involved, and we love to have people visiting our website, interacting with our online tools, and even paying us a visit. So we don't only work on malaria and HIV, as Caitlin mentioned. We actually work on a plethora of diseases, many of the high burden diseases that people experience today. And so we use generally the same model for all of these, but they're adapted to the biology and the epidemiology of each specific disease. So you can go to our website and you can see all the publications that we've produced, some of the modeling results, who we are, our backgrounds, and our partners. So we don't work alone, obviously. We work with many partners in Seattle. The Gates Foundation is one of our big partners, but we also work with universities across the world, other institutes. And so what can you do? So you can check out our website. You can actually download our software. It's free. It's meant to be developed and then given away. I will warn you that it's very difficult to use, so you probably won't be able to use it. But at least you can see kind of some of the kind of nuts and bolts if you go to our website on how it works. Yes.
We've put a lot of effort in recently to make it more user-friendly. There's documentation now, which is...
HIV model is really hard to use, but maybe the malaria model is easier. So yeah, if you are interested in getting involved in this, if you are a person starting their studies, in the middle, at the end of your career, whatever, we really encourage you to get involved. What if I want to be a disease modeler? I never thought I would be doing this professionally. I went to school for environmental science and got really interested in geography, and that led me to the geography of disease, and then epidemiology, and then disease modeling. But people come from a variety of backgrounds. So Caitlin has her PhD in biomedical engineering. Is that right? Okay.
But I started in astrophysics, so it's like...
We have actually most of the people who work at IBM are physicists for some reason. So I'm one of the two, I think, actually bread and butter trained epidemiologists. I got my PhD at University of Washington in epidemiology. But there are a lot of ways to get involved regardless of where you're at. You don't have to have a PhD or even a master's to be involved. We have a summer internship program where we even have a high school student who came. He was really interested in learning about modeling. And so there's a lot of opportunities for people. So you can seek us out online. And people come from a variety of backgrounds, like I said. So, if you're starting your studies and, you're going off to college, do what you love. Study Portuguese, but maybe take a math class here and there, because it might come in handy down the road when you start to figure out what you actually want to devote a lot of your time to. So thank you so much for your attention. We're going to take a little break and then pass it over to Brett.
Okay, so for those who are still with us, I ask that you guys re-find your seats. I'm going to ask Caitlin and Adam to once again join me up here on this stage. At this point, if you have questions that you would like to ask of them, please do. Raise your hands, I'll call on you. Also, ask anything. So here, I'll pass it over to you guys. Anything.
Yes.
Okay, so I don't know if this is two-part or not, but like with your small area simulations, how do you deal with people moving in and out, or like if a neighboring country doesn't apply all of the techniques, how do you deal with like the next state over is only applying one of the techniques, and so it's just going to come back?
So those are all very, very important questions. I didn't show this in the simulation, but in fact we do include migration in the model. So we do our best to try to understand how people are moving about the region, and we'll also try to understand how mosquitoes might be moving about the region. And we need to be very careful too about understanding if people are traveling in and out of the area and maybe bringing infections back with them. It can be quite tricky because you need somebody to collect data to inform that, and oftentimes it's just not available, so you have to make your best guess. But yeah, it can have a serious impact on what your conclusions are.
Would you have to apply all of the things to all of the countries to actually get it to work?
No, not necessarily. So for example, what we might do is we make some bulk assumptions about the area that we're studying and how many people are coming in with infections from anywhere.
Yeah.
So you model some sort of external place and you say, people are coming from a region, they're going over there, they're coming back, and a certain fraction of them will be carrying infections. But we don't necessarily care if they went to Mozambique or Democratic Republic of Congo.
So if there's international or civil conflict, does that just throw off the whole model and well, so much for 2018?
That is a huge issue because especially in... Okay.
Thank you.
Yeah. So the question was about, dealing with things happening across borders, and in particular, if you have a civil unrest in one country or a refugee crisis, how that might affect the disease in the neighboring countries. And this is something very important because for malaria, at least in sub-Saharan Africa, the different countries have very different approaches. The governments have sort of very different levels of commitment So Zambia is an extremely committed government. They've committed a lot of funds to solve this problem, but they neighbor the Democratic Republic of Congo, where things are not going real well right now. There's supposed to be an election, but we'll see if that happens. And there are a lot of cases that come across that border, which might be aggravated if the refugee situation becomes worse. But there's really not a lot you can do about that. And the other issue is that mosquitoes don't understand borders. So mosquitoes fly through, they don't show their passports. So you just have to do your best to understand what's happening, to figure out, if you have to step up your interventions to deal with that, how much do you have to worry about it?
How much crosstalk is there between these different massive public health problems? I mean, if you gang together the results of your malaria model with your HIV model and considering the ensemble of disease in the same region, how much do those play against each other?
Okay, so it depends on the combination of diseases that you're talking about, but yes, it would be wonderful if we could take advantage of all that we know and all the approaches that we have to deal with multiple diseases at the same time. I know the Bill and Melinda Gates Foundation, for example, is trying really hard to implement cross-cutting approaches. So trying to find solutions that have impact in multiple disease areas. So for example, in Democratic Republic of Congo, just bringing up again, both malaria and sleeping sickness are very big problems. So they're thinking about designing a diagnostic test that can test for both of those diseases. So that's some way you could encourage people to get more testing. You can save some money by not having to deal with two different programs. But it's hard because oftentimes disease areas are quite siloed. Each disease area has its own conference. This is an issue, but it would be great if we could work together more.
Some of the interventions actually do overlap. So there is a prophylactic antibiotic that is given for HIV patients to prevent them from acquiring what's called opportunistic infections. So these are infections that manifest in people whose immunity is very low, and so they'll take a daily antibiotic. That same antibiotic actually protects against malaria. And so there are these sort of interventions that do cross-cut, but then there's also these kind of more root causes like poverty and income inequality, sexual violence, these kinds of, you can get even further back and improvements in terms of water and sanitation. These are big interventions that can have cross-cutting results.
So the other thing is that there are a lot of data sets that would be useful to researchers and modelers across different disease areas. So for example, this might sound trivial, but knowing the population of a country and where people live is sometimes difficult to know. But that's something that's really key for people to understand what's happening in malaria and HIV and polio and all of the diseases that we study.
My friend and I have a question that's specific to the HIV slides. That's more an observation with a question mark. But when you were visualizing the spike in the virus and then the drop-off, and that it was the antibodies in the immune system that causes that drop-off, and that when sensing HIV, you look for those antibodies. If it's that peak where most of the infections happen, are you looking for ways to preempt that peak in preventing HIV spread, or is there anything like that in your modeling to handle?
Yeah, so that peak happens pretty quickly. So the question was, if I can summarize, let's see. So the peak in viral load occurs early, and that peak is a predecessor to the then peak in your immunity, which I didn't show. But the virus replicates in your body to the point at which then your immune system, your CD4, your T cells, start to detect it and produce antibodies to it, and then attack the virus, and then that's what leads to the downfall. But the question was, could you preempt that peak in some way? And it's very difficult because even before people are even aware that they're positive or that they're infected or they have symptoms, or they think they have the flu, let's say, that's when that peak is happening. And so it's very rare that someone is going to get an HIV test before that peak happens, because that peak is sort of when the symptoms are developing and at their strongest. So it's very difficult to preempt that peak, and I don't think there's really, unless you test you just have a very high risk population where you test weekly. So I think that is something that HIV modelers are concerned about, and there's actually a big debate in the field about the role that's called acute HIV infection, so the early phase. What is the contribution? Because it's really short. So something with modeling is that you have to understand, that we try to understand, is that you have a lot of virus in your body. You're at risk of transmitting, but it's a very short window of time compared to people have HIV for years, and so in fact it's a huge contribution per unit time, but as a whole there's a debate about how much it actually contributes to HIV onward transmission. So I think it's a huge deal, and I think that the only way to really stop that phase is to stop people from getting infected in the first place. So that's where there's not really treatment's not going to really be an effective intervention for that. But that's where you have to sort of say, okay, we can prevent so many HIV infections, we can't prevent all, and so we have to pivot to other prevention methods, and that's where kind of this combination HIV prevention, like Caitlin was talking about, there's not really one silver bullet, but if we do treatment and we do prevention, that hopefully would take care of it. Let's see, way in the back.
So I have a question about the variables in your that you used for modeling. So when you do your models, how can you attribute to what the success of how can you attribute to what the success is that actually happened in your model? Because like you said, you're using a lot of these traditional prevention approaches. But we have condoms at every gay bar here in Seattle. But that doesn't mean that HIV doesn't happen. It's this reduction of stigma that probably can come to be attributed to the diminishing of HIV in the United States. So how do you put that into your models to attribute the success not only to just condoms but also to advocacy that goes into this?
That's a really great question. So the question was, how do you attribute the effect of an intervention to the actual intervention and maybe not something else? So for example, if you have condoms available for HIV prevention, how do you know that it's that availability of condoms and not they're also at the same time trying to have stigma reduction campaigns simultaneously? And that's actually where modeling becomes very, very tricky. And there's a classic quote that all models are wrong and some models are useful. And so it's really, it's similar to climate models, or anything that, predicting the weather. Human populations are very complex, and human behavior is very complex. And I think the goal of modeling is not necessarily to be able to depict every single intricacy of human behavior and society and disease transmission, but kind of the major forces. We have a lot of parameters in our model, so we might have a condom parameter, and we have to estimate, how often are people using condoms, and what's the frequency, and who's using condoms, and all that kind of stuff. And that comes from the availability of data, but sometimes we don't have data on that, and so we have to just give our best guess. But sometimes you're right, we're modeling the effect of condoms, but in fact it wasn't the condoms that were doing it, it was another educational campaign. And so that's really where the critiques will come in, and people will refine the models, and that happens constantly. So people are constantly refining the models. In this example, an example that I gave above, the early model said that test and treat was gonna make the epidemic crash, but it didn't consider that there was this heterogeneity in who was being put on treatment. And so, I'm trying to build on models like that to say, well, we need this extra level of complexity, but you don't want to get so complex that you can't interpret the outcome. So it's this balance between complexity and something that makes sense.
So my question to follow up to yours is, at any given time, be it, and you can correct my vocab here, how many parameters, variables are you looking at in any given model, and when do you say, oh, that's too much, right? Like, talk a little bit about what goes into your model.
So I think the answer to that varies widely across both the disease areas and the questions that we're trying to answer. So for example, in the malaria space, we have several versions of the model of different complexities, depending on whether we need to answer something very, very specific or whether it's something more general. Say a question that's related to, individual people versus the entire country.
Like the one that you showed us.
Hundreds, probably. But what I didn't really go into when I was saying what a model, like what the model development process is, as Adam mentioned, it's iterative. So you start by building part of the model that you have data for so that it, does a good job of reproducing the data you've seen. Now, whenever a new data set comes along, you have to make sure that it still can reproduce all of the data. If you're interested in asking a new question, a new epidemiological question or biological question, you have to make sure that all components of your model still work. So the stuff that you've already built, and also the new pieces. So the malaria model, somebody started building it 10 years ago. And it's been growing piece by piece ever since. But you have to make sure all along that everything is right. So if you start by building, how parasites are developing in the body, and then you add on, like how parasites are getting transferred through mosquitoes, and then you add on, people are moving from place to place, you have to make sure that everything is still working all together.
Yeah, we have hundreds of, pretty much the same as Kayla's. But we have hundreds of parameters, and some of them are the same. So we have a parameter that's the probability of an infection from one coital act, so one sex act between a positive and a negative person. We set that as a probability in the model. It's 0.002, I can tell you what it is. So it's very low, but with the number of sex acts, you have infections. That's just one of probably, I would say, 500 parameters. Only 30 of the parameters are we tweaking there's a fitting process. So we calibrate the model. So what we do is we have all these parameters, and then we run the model, we see how close it came to the hitting the prevalence that really actually occurred, the data. And usually when you run the model for the first time, it doesn't do well. And the model will tweak itself. It'll change I have 30 parameters right now that change every time. It changes a little bit, runs it again, changes it, runs it again. We do that thousands and thousands and thousands and thousands and thousands of times, and then each time you run it, you get a parameter set. So you get a set of values, and then what we do is we try to pick the best set of values. So the question from the gentleman up there, you can get a set of values that really reflect the epidemic nicely, but are the wrong set of values. So you can have many, many, many different parameter sets that give you the same result, and that's a problem. Okay, so we have 4 questions. So can we just do this in order of closest to me to farthest away from this time? So the gentleman in the front.
The malarial district that you were modeling looked to be a bit removed from the lake, and so you saw seasonal variations in malarial infections. I'm curious, is there a district closer to the lake, and would it be less seasonally dependent, and would the interventions be the same?
So you know something about malaria, I'm guessing. So yes, we have in fact modeled areas right along the lakeside as well. And they do differ somewhat from what we see in Wenyuwe. in large part because they have different types of mosquitoes. Yes. So it is true that it also looks a little bit less seasonal because they also have the mosquitoes that you have in Munyo Way, but then they have a second major species of mosquito that is more active in the dry season. So you have to worry about one type of mosquito for half the year and then the other type of mosquito for the second half of the year. So that means that solving malaria in those places is much harder. But I will add that they threw the malaria kitchen sink at these places too, and they also came very close to zero. The tricky bit is that they're seeing the malaria come slowly back in those places. So not only do you have to work hard to get rid of malaria in those places, but you'll also have to work really hard to make sure that it doesn't come back.
Let's go to the next, yeah. Great.
Okay. I just wanted to ask you what sort of algorithms you're using. It sounds kind of like regression, but how is the model learning? What's that model?
So we have a few different optimization algorithms that we use, and this is something that has been evolving over time. So in addition to the people at IDM who focus on the research areas and the software engineers, we also have a team of people who are basically doing math as their job, and a lot of them are focused on this problem of how do we calibrate a model and what is the best optimization algorithm to do that. So, I mean, a lot of it is quite traditional. If you're an expert in optimization, so for example, you compute a likelihood function which compares the relationship between your data and your model, and then there's a function that updates the parameters. You test those, see if you improved the likelihood function or if you made it worse. If you made it better, then you proceed in that direction. But we have a number of different ways that we're doing that. It's we can talk about specifics afterwards if you like.
Yes.
I assume you've had the opportunity to see how well your model predictions fit the reality. Make a prediction and then come back 10, 15 years later and see how it does. Have you noticed that the predictions are systematically high or low, or?
So I don't think we actually have a ton of opportunities to do that, because if we're tasked with making a recommendation about what you should do, they're going to do that and not the other thing. So it is true that we might make a recommendation to do something and things just don't go well at all. But generally, we make recommendations that are pretty sound. So when you make a recommendation, you have to think carefully about how certain you are about those predictions. And if you're not very certain, then you're probably not going to make a recommendation at all. So you make a certain prediction, it's quite likely that that's going to happen. And the people implementing the programs won't know what would have happened if they'd done something else. Two more, please.
Yeah, I was curious a little bit about the process that the organization uses to develop models. So, for instance, obviously, some of it is obvious. It would happen anywhere, but some of the parameters might be a surprise unless you have people actually in the area studying. So I'm curious, how do you do that kind of study?
So, okay, I'm going to try to repeat the question. So, question is about, how do we come up with the values for parameters when sometimes it's difficult to do? Is that sort of like a, do you want to rephrase it again?
Yeah, so obviously you're gathering requirements for how to build these models, and I'm just curious how you gather those requirements. Do you have people that go to the areas and study behaviors and what kind of people are involved in doing that kind of thing?
Okay, so I guess it's, how do we get the data that we need to calibrate our models? And what kinds of people do we work with to do that?
Yeah, and obviously you can't do that kind of study in a vacuum. So I'm curious
right.
how you
So we are definitely not alone in our disease areas, right? There are a lot of people who do field studies and they publish papers about these things. So a lot of what goes into the model will be based on publicly available data that comes in scientific journals. So we also have some capacity in some very special cases to be involved with trial designs. And so we can tell people who are working in the field, we'd like you to collect this certain piece of information. So in Zambia, for instance, we had a little bit of a chance to do that. To be like, this is really important for our model, so we'd really like you to add this question to the survey when you're going door to door and doing mass drug administration. But I don't think that too many of us are actually out in the field collecting the data ourselves. It's more that we have relationships with people who are collecting the data, and we can make specific requests to help us with the models.
So for the HIV field, we work with a lot of partners who are on the ground. We have a relationship with an institute in South Africa, outside of Durban, where the epidemic is really some of the highest in the world. And they collect they do these huge surveys. So we rely on a lot of survey data. So people will go out and they will survey huge swaths of the population and get a lot of information on healthcare utilization, demographics, and even things about like sexual risk behavior, which is really important for the HIV model. One particular piece of information that is really useful for our HIV model is how people form relationships and dissolve them, so that the duration of time that people are in relationships is really important, whether they have extramarital or extra primary partnerships. Relationships outside of the partner. These are oftentimes really hard data to get because they're very sensitive, but we do have some data coming in about how people form relationships, and then how old your partner is a huge driver of HIV risk. So people thought that it was young women with much older men that was driving the epidemic. Our model is showing that that's actually not a huge driver, and we actually have done some primary data analysis to show that it's not those types of relationships are actually they're risky, but they're rare, and so they're not a huge driver of the epidemic. Was there one question? Yeah, right next.
Somebody brought up earlier a question about if there are reservoirs of any of these diseases that are not human, and so I wondered if you could comment about how that changes the modeling process for something like tuberculosis, where there are non-human reservoirs that may be quite important. What kinds of data are collected that are different in that sort of situation?
So I think the truth is that we IDM have not done a lot of modeling of zoonotic diseases. So I do a little bit of work in sleeping sickness, and in sleeping sickness, it's definitely possible for the trypanosomes to live inside cattle or lizards, or they've actually found it in all kinds of different animals. And so if you're thinking of eradicating a disease, of getting rid of it completely, you need to think very, very carefully about what's going to happen to those parasites in those different animal species. So we've thought about these issues, but we haven't yet implemented it in the model.
Yeah, I used to work on a disease. I was at UC Berkeley working on a disease called Schistosomiasis, which probably most people haven't heard of, but it's one of the most prevalent infectious diseases in the world. There's about 200 million cases globally, and it's transmitted as a very complex life cycle, but it's transmitted partially by a snail, and then also has a mammalian host, and it can be harbored in humans, but also cattle. There's a rat form of schistosomiasis. There's a duck form of schistosomiasis in Green Lake that actually, but humans can't get it. It's called swimmer's itch. Yeah, it makes modeling much more complex, because then you have to bring in a vet, and then you have to take samples of feces from cattle, and it makes the process a lot more fun for epidemiologists.
Just to add to that, so a disease you might have heard of is guinea worm. And so the Carter Center, for example, was thinking that they were very close to eliminating guinea worm, and then it was discovered that there are a lot of dogs that carry guinea worm, and they're transmitting them back to the human population. So we haven't explicitly modeled this mechanistically, but there is someone at IDM who did some work to try to understand where you are likely to find dogs that are likely to get infected. So not everything that we do has to be like a complicated model that involves all aspects of the disease or the system. Sometimes we can come up with simple statistical relationships that are still helpful for telling people what they should do to try to solve the problem.
And I think we have time for one last question, and it'll be over here.
I just have mine's kind of a multiple step question. Comes from just the area we live in and the interaction between all of PATH and CID and Fred Hatch and stuff. I'm coming at it from looking at what you're doing to explain to students what you do. They come, they see what goes on at PATH. Maybe they go to a field trip somewhere at CID and they learn about malaria, they learn about global health, and then they learn about an organization like yours. And I kind of went, which came first, the chicken or the egg with you guys? Where do you fit into that? Are you getting your modeling questions, your modeling data? Is it being driven by particular organizations within that collaboration of all these places that are studying similar things? Or are you independent? I'm kind of curious how you fit in with all of those other organizations that are not only gathering data, but they're on the ground, they're doing their own research, and their research is something you're drawing from, but how much do you also influence, like you said, the data gathering that they're going back to Tanzania, maybe the CID, and getting stuff from the mothers that they're working on an ongoing basis? How much integration is there between, where did you come from? And how much are you integrating with some of these other things that my students hear about? When I present to them, this is organization I came to listen to a lecture about, how do I explain to them your interaction with those other organizations? Is that a nice complicated question?
That is a great topic. So IDM has a pretty close relationship with the Bill and Melinda Gates Foundation. So we are funded by Bill Gates through the Global Good Fund. We are not part of the foundation by design. So the idea is that we can do our work independently and answer their questions without being influenced by, whatever ideas they have internally about what should be done. So they have to answer a lot of questions about how they spend their money and how they should evaluate the projects that they're funding. So they might ask us to do a modeling project to sort of confirm or deny their suspicions about something, or to say, we have these 4 different possible interventions that we might invest in. Which one do you think makes the most sense for us to put our money on? And then we also interact with a lot of partners who are grantees of the Bill and Melinda Gates Foundation. So a grantee will present the work that they've done to the foundation. The foundation will say, well, that's very interesting, but we think maybe you should talk to the people over at IDM, and maybe they can do some modeling to help you figure out, say, how best to design your next study. So then that organization, say PATH, might come to IDM explicitly, and we'll work with them very closely to figure out how to address that issue.
Yeah, so questions come from a lot of different places. So yeah, there's the Bill and Melinda Gates Foundation that sometimes will feed us questions. But the kind of beauty of IDM is that we're not beholden to we're not beholden to grant cycles, so we're not like an academic institution where we're spending half of our time writing grants. And so we answer very specific, so I was working on a project where the questions were coming from ministries of health in sub-Saharan Africa who wanted to know what the effect of huge circumcision campaigns was going to be, because circumcision is actually a very effective HIV prevention strategy, and it's being scaled up in a lot of countries that don't traditionally practice it. And so they came to us, through the Gates Foundation, they came to us and they said, well, can you model this and give us some prediction of, if we circumcised 100 men at a certain age, how many HIV infections would we expect to see prevented by 2030? And it ended up being a cost-saving strategy for them because they didn't have to put all these people on treatment. Not only was it cost-saving, but it saved people's lives. But we also get to work on very fundamental questions about disease transmission and biology that universities don't really get to work on because they're sort of high-risk, high-reward questions. And so not only are we answering very fundamental questions from the HIV community, but we're also kind of responding to the literature and saying, wait, this doesn't make sense. Let's simulate this and let's do some more kind of modeling with more intricate, more complex data. And so we really get to be a laboratory in that sense and try to answer questions that the HIV community or the traditional HIV communities is not able to answer because they're beholden to certain funding agencies. But we like to be integrated. We want to be partnering with a lot of people. We want to be sharing our software. So a lot of times a university will come to us and say, or a PhD student at University of Washington or a master's student will say, I have this project, I really want to add a component to my dissertation where I want to look at what the effect is going to be. And then we will take them on and we'll say, we will provide support for you for 6 months and get you up to speed. And then they'll, the idea is to really disseminate our model as much as possible because every time someone uses it, it gets better.
So at this time, I want to say thank you to Adam and Caitlin for their talk tonight and their availability. I think their talk was amazing, and I think from the questions that you guys had, you felt as well, so thank you guys. I do have my own last question, and if it's still here, what's that? What are we seeing in Washington right there?
Okay, so this is actually related to the idea that that whether or not you seek care for your illness is a function of where you live. And when we do these calculations, we start with a map that looks like this, that tells you how hard it is to get around a particular place. So for example, if you're in the mountains, it's really hard to get around. So if you want to get from the mountains to the nearest hospital, that's something that you have to account for. So this is just a map that colors the state of Washington according to that difficulty of moving around.
Okay, I was very interested. I read the colors, but I didn't know what it was about, so thank you very much. I do want to use this as a point to jump off, and we can go this way, and to remind you that we do have other talks. I want to say with the Global Health Series as well as what we have with the Innovation for Good, we're actually very much expanding on a lot of the ideas that Adam and Caitlin talked about tonight. So if you want to hear more pieces of the puzzle that go into kind of figuring out what parameters they put into models, or if you want to hear about the whole holistic approach of how disease is looked at and taken care of, I highly recommend you guys coming to our Global Health Series as well as the Innovation for Good that we have later on. And with that, I just want to say once again, thank you all so much for joining us tonight. We have a couple more minutes if you want to come down and personally chat with Adam and Caitlin at this time. And then I also say thank you very much for filling out those surveys, because guess what we do with our surveys? We evaluate and we actually don't necessarily model, but maybe in time. So that you will all become little data points in a model that we might develop later in time. So thank you all once again. Have a good night.